Hello, how you doing, folks? Here it is, April 20th, 420. Here is my 20th lecture, Baked, the science behind cannabis edibles. So before I launch into this lecture, I need to offer up some disclaimers. First of all, I am not endorsing the use of cannabis, components of which are classified by the U.S. federal government as Class 4 narcotics. Secondly, I'm just presenting the science behind the consumption of phytocannabinolic compounds, because understanding some of the science behind it can perhaps inform policy decisions, can inform personal decisions. It's just better to know the science behind this movement that is certainly spreading in popularity. And then lastly, I will simply read from you this passage from the Colby College Student Handbook. Students are prohibited from possessing, using, furnishing, or otherwise selling recreational or medical marijuana or cannabis extracts not approved by the FDA. Maine law that permits the use of recreational marijuana for persons 21 and over and medical marijuana with appropriate certification do not apply on Colby campus, in Colby vehicles and facilities, or at Colby events. With that said, I'm simply going to start by saying cannabis edibles are becoming mainstream. What used to be something that was hidden from view and a social taboo, society has moved significantly towards liberalization on this issue. For example, here is a restaurant in the Lowell Farms Cafe and Herb Company in West Hollywood, which purports to be a farm-to-table restaurant for eating gourmet foods and drinks that have various cannabinoids, either CBD or more psychoactive components in the food. It's pretty mainstream now. So, and this is the fact that it is not mainstream is not necessarily something new. The use of cannabis goes back to ancient pre-Neolithic civilization. And it's probably, so cannabis was probably the first, if not one of the first, if not the first, crops to be cultivated by humans. They have concrete evidence of its cultivation going back at least 10,000 years. And it was used for the fiber to make rope and cloth. Food, the seeds are particularly nutritious. I am not going to spend any time talking about the protein and fat content in the seeds, but they're really a superfood, if that term has any meaning at all. It should certainly apply to hemp seed. And then, of course, the psychoactive uses of cannabis for both recreational and spiritual use. And there's all kinds of evidence throughout that time span between the earliest record and today. So, for example, this is a depiction of bong eaters. Bong is an edible preparation of cannabis that has been consumed for 3,000 years in the Indian subcontinent, which is the point of origin for at least one of the two major strains of cannabis. Here's the Chinese character for hemp that depicts two plants being grown under a shelter and alludes to the long-time cultivation of this plant in Asia. So, the psychoactive component of cannabis is THC, and THC and the host of other cannabinoids. We'll see in a moment that there are lots of them. were probably the plant's evolutionary defense against insects and UV light. The THC itself was first isolated by this guy on the left-hand side here, this Bulgarian-born chemist Raphael Mechulam at the Weizmann Institute in Israel back in the 60s. And you can see one of the buds on the hemp plant here with the trichome, which is the little drops of the cannabinolic oils decorating the surface as a defense against insects. Insects don't really want to eat this stuff. And whether there's been a coevolutionary process with humans is certainly debatable. The endocannabinoid pathway in humans is not exclusive to humans and is found throughout at least mammalia, if not the animal kingdom. All right, so there is a remarkable structural diversity in the cannabinoids. These are just two of the most commonly known cannabinoids out of at least 144, perhaps a much larger number than that, found in cannabis plants. So on the left is delta-9 THC, tetrahydrocannabinol. This was discovered in '64. And then CBD, cannabidiol, is a cannabinoid that was actually first isolated back in 1940. Neither of these compounds, the delta-9 THC nor CBD, are actually psychoactive at all, and the majority of the cannabinoids are not potently psychoactive. Delta-9 THC can become converted to a psychoactive form by the liver, as we'll see later in the lecture. However, not cannabidiol. A lot of these cannabinoids have all kinds of medicinal and anti-inflammatory properties, sort of alluding to the long-term acclimation, so to speak, of human physiology to this plant and the chemicals that it produces. On the left, we have tetrahydrocannabinolic acid. So you'll notice in this structure that there's a carboxylic acid group appended to that phenolic ring there, that aromatic ring of the alternating double bonds, six-membered ring. This compound is non-psychoactive, and it is by far the largest component of any of the cannabinoids found in cannabis. It is not psychoactive. However, it can go through a process called decarboxylation, which we'll talk about, where that acid group is actually removed and the molecule is converted into delta-9 that you saw on the previous slide. This compound here, tetrahydrocannabinolic acid, is been shown in fairly rigorous clinical studies to have anti-inflammatory properties, to also be neuroprotective, so to protect the loss of neuronal density in the brain with time in aging. It is also an antiemetic and suppresses nausea, so it's often used for people who perhaps are going through chemotherapy or something that's suppressing their appetite. It helps them maintain that appetite. And it also affects metabolism in the liver and can help prevent fatty liver disease. It affects lipid metabolism in the liver, and because of that, it is an anti-obesity compound. On the other hand, here we have cannabichromene, CBC, different than CBD. This molecule is anti-inflammatory as well, and there have been studies that have shown that it contributes to blocking the nociceptive pathway or pain pathways in the body. not working through the delta opioid receptors. It does promote neurogenesis, meaning the growth of new synaptic connectivity and the growth of new neurons in the nervous system. And it is entirely non-psychoactive. So these compounds are often mimicking the effects of molecules in the body called endocannabinoids. These are molecules that the body makes that serve various purposes in the nervous system and the immune system. We're going to take a quick look at that now. So the first of the molecules that I want to look at is this arachidonyl donoyl glycerol, 2-AG is how I'll refer to it from now on. Highly concentrated in the central nervous system. If you look at this molecule, it's essentially a monoglyceride of the the omega-6 essential fatty acid, arachidonic acid. We've seen arachidonic acid in the chapter on lipids, and this is just the two substituted monoglyceride of arachidonic acid. This is an endocannabinoid, and it attaches to this. It is the ligand that activates this receptor on the right-hand side, this membrane-bound receptor found in neurons in the central nervous system, the cannabinoid receptor 1, CB1. So this is a little picture, a ribbon diagram of that receptor that would be embedded in the membranes of neurons in the central nervous system. Another of these endocannabinoids is called anandamide, which is the short name for N-arachidonyl ethanolamine. It's a fatty acid neurotransmitter. So it's basically the amide adduct of arachidonic acid with ethanolamine attached to it on the end. This name, anandamide, it comes from the Sanskrit word ananda, which means joy, bliss, and then the word amide. So it's the amide of bliss. We find this molecule in basically all tissues in a very wide array of the animal kingdom. It's also found in plants, including notoriously in chocolate, and has been implicated both the presence of anandamide and other N-acetyl ethanolamines, which have similar structure but without having the arachidonic acid tacked on the backside. Those other acyl ethanolamines are responsible for preventing the breakdown of anandamide in the body, and has been implicated in leading to the pleasant chocolate high that people have. So let's take a closer look at some of these receptors in the endocannabinoid system. There are two of them that have been the most widely characterized so far. And these two receptors, CB1 and cannabinoid receptor 2, CB1 and CB2, the effects of CB1 and CB2 can be modulated by their combination with other membrane-bound proteins to affect the specific signaling pathways that they are able to affect. We're going to see a little bit of that in a moment, one example of that in a moment. But CB1 is, both of these are found throughout the body, and so these are just some generalizations about where they're more concentrated. CB1 is found primarily in the central nervous system, the brain and spinal cord. It's going to affect things such as appetite, motor activity, and coordination. CB1 can have neurogenic or neuroprotective activities, and it affects synaptic plasticity, so basically learning the remodeling of synaptic connectivity in the brain. It's been implicated in several aspects of hypothalamic function. The hypothalamus is the organ in the brain, the part of the brain that sort of overlaps and connects the nervous system with the endocrine or chemical signaling part of the body. CB1 has been implicated in pain perception and aspects of short-term memory formation and cognition, basic cognition or thinking. So it does a heck of a lot. It modulates all of these different functions. CB2 is more concentrated in the peripheral organs and the peripheral nervous system, and also implicated in, it's also found in a number of cells in the immune system. It can suppress pro-inflammatory cytokines. So cytokines are hormones that cause action in various immune cells in the body, and it can suppress, CB2 can suppress the production of pro-inflammatory cytokines. It's been implicated in bone mass regulation, the cardiovascular system, eyes, the GI tract. It's anti-inflammatory in the GI tract. Can lead to metabolic balance in the liver and pancreas, kidneys, the reproductive system, on and on. CB2 is also found in the skeletal muscles and related to the skeletal muscle stamina. Some have suggested that that activation of CB2 is actually, rather than endorphin release, it's CB2 activation that is responsible for the runner's high. So all of these things, pretty important receptors. So to understand how phytocannabinoids, or the cannabinoids from cannabis, affect all of this, we need to understand a little bit about neuronal signaling and a little bit about how endocannabinoids work. So here's just a primer on how neurons work in the nervous system. We have this diagram with two neurons that are going to signal. On the left-hand side, there's the upstream neuron 1 that's going to send a signal down to neuron 2 at this junction called a synapse. Along the axon, the signal comes down the axon to the presynaptic terminal, and then this comes into contact with the dendrite on the downstream neuron in neuron 2, where there's a postsynaptic terminal that's coated with these various types of receptors. There's also a couple ion channels. These are doorways that can open and close in the cell membrane to let either calcium or potassium flow. There's also sodium channels that can allow these ions to flow in or out of the cytoplasm of the cell. And then lastly, you'll see there are these vesicles, basically little membrane-bound bubbles that contain neurotransmitters that are going to activate the receptors on the downstream neuron in the dendrite. So a signal comes from the upstream neuron in the form of an action potential. That causes these voltage-gated calcium channels to open up and allows calcium to flood the cell. And that causes membrane fusion of these synaptic vesicles with the presynaptic terminal. Once that vesicle fusion happens, they release all their neurotransmitters into the synaptic cleft, that area, that space in the synapse between the presynaptic and postsynaptic terminal. And those neurotransmitters can be a host of different neurotransmitters. They can be excitatory neurotransmitters, small molecules like glutamate, the conjugate base of glutamic acid, the amino acid. There's inhibitory ones like gamma aminobutyric acid, another signaling molecule that turns cells off. It just depends on the specific synapse. There are all different types. So once the receptors get activated, these are chemically gated receptors, it causes an influx of calcium in the postsynaptic terminal, which leads to anterograde or normal signaling that happens in the cell, and it changes the voltage in that downstream cell, and there's like a signal that progresses through the neuron. nothing about endocannabinoids yet. However, that increase in calcium in the postsynaptic terminal starts a process where arachidonic acid that is embedded in the membrane, because it's in all membranes, it's just a fatty acid. Arachidonic acid gets converted to one of the two endocannabinoids that I talked about, anandamide or 2-AG. And those then get released from that postsynaptic terminal. Those endocannabinoids are going to travel backwards as a retrograde signal, a backward signal across the synapse to activate the cannabinoid receptor on the presynaptic terminal. So you can see here that that receptor that I showed you several slides ago is embedded in the membrane of the presynaptic terminal. And these endocannabinoids that our body makes are the ligands. They are the thing that binds and activates those receptors. Once that binding happens, activation of that receptor leads to the stimulation and formation of a second messenger, this small molecule called cyclic AMP, which causes a host of different signaling cascades to engage. One of them is the closure of the calcium channel and the opening of a potassium channel that allows calcium to flood out of the cell. This affects, basically slows down the degranulation or the release of the neurotransmitter into the synaptic cleft, and sort of attenuates, modifies that signaling event that's happening at the synapse. Because there is now a difference in receptor activation and calcium flux that is going to have an attenuation or a dampening of the endocannabinoid retrograde signaling pathway. This is an example of what's called negative feedback homeostasis that's happening. It's this sort of negative feedback thing where the signal that's turning down the effect of the signal itself modifies, and it's going to find in equilibrium with time. So now let's introduce the phytocannabinoids. As I said, there's about 144 different naturally occurring phytocannabinoids that are going to interact with one or other of the cannabinoid receptors. We got to consume these somehow. We have to get them into the body, either orally, inhale them, sublingual, dermal patch. Man, there are so many different ways that people ingest this stuff. But anyways, when it gets in the body, it is able to attach to the same endocannabinoid receptors and also affect the cyclic AMP levels, increasing cyclic AMP signaling. That is in turn going to have similar or at least analogous effects to the endocannabinoids. It's going to close down calcium signaling, open up potassium channels, and further modulate neurotransmitter release, and further effects on the retrograde signaling pathway. So the effects of all this is just one example. The effects of all this can vary widely depending upon the phytocannabinoid that's being consumed, and the specifics of the synapse, the neurons that we're talking about. All right, so let's look at the strength, onset, and duration of the various effects of cannabis, the effects versus the cannabis consumption route. So I talked about how people inhale it. It can be consumed orally, or it can be placed under the tongue. Inhaling it, the two ways of inhaling it are smoking or pyrolysis, and then vaporization. So this has very fast onset and a strong effect peak, but it's very transient. It rapidly declines, and leading to a short duration of the experience. Whereas an oral consumption these are edibles, can also be beverages. There's going to be a delayed onset. It can take anywhere from a half hour to up to 2 hours before it really affects you, depending upon what else is in the stomach, what other food these were consumed with. So there's a very drawn out effect peak and a gradual decline in effects. This overall leads to a very long duration of experience. And the danger here is one of them is that is dosing, because the onset is so delayed. It's hard to know whether the person has consumed the amount that they were hoping to consume for perhaps medicinal or recreational purposes. So it's easy to consume more than a person had intended. THC, they don't even have a lethal dose established for THC. So really what can happen is you just have sort of the unpleasant experience. If you consume too much, you have an unpleasant experience of having more in your system than you want, but it is transient. It will go away and will not leave any profound long-term effects. And then the third method of consumption is sublingual absorption. So this is, you can melt an individual can melt edibles under their tongue, or take various tinctures extracted often into ethanol or something like that. It's a mid-range onset, drawn-out effects peak, and a gradual decline in effects. So in general, sublingual absorption is pretty similar in effect to vaporization. Inhalation, there's a real difference between smoking or pyrolysis and vaporization, because smoking or pyrolysis actually combusts a lot of the THC or other cannabinoids that are in the flower, and leads to a less efficient absorption of the molecule, which is one of the reasons it has such a short effect. Whereas vaporization, you get a broader range and less destruction of the molecules entering the system. So several transformations must happen for THC to be psychoactive. The first of those is that tetrahydrocannabinolic acid, THCA, has to be decarboxylated into the delta-9 THC using either heat or light. And THCA is completely non-psychoactive. As I said, it's the predominant THC found in cannabis. A lot of people consume raw cannabis looking for the medicinal properties of THC. THCA is not psychoactive because it does not bind to CB1, that carboxylic acid that you see sticking off the structure in the middle of the THCA prevents THCA from getting in the binding pocket of CB1R that I showed earlier. But if you decarboxylate it with heat or exposure to sunlight or UV light, that it releases carbon dioxide, and it turns into delta-9 THC, which itself is very minimally psychoactive. It's not strongly psychoactive at all. It will bind to CB1R, but very moderately. However, once delta-9 THC is in the body, once it gets to the liver, it can be converted to 11-hydroxy THC, and you'll see the addition of the hydroxyl group at the 11 position there. This is the main psychoactive metabolite of THC that is responsible for the powerful effect that is seen with various edibles. And you'll remember that one of the main jobs of the liver is to remove various chemicals from the body by making them more soluble. And so it should make sense here that the liver is sticking a hydroxyl group on this molecule to get it more soluble in water so that the kidneys can excrete it into the urine. All right, so I want to untangle the reason that edibles have the different time course and physiological effects that they do compared to the other modes of ingestion. But to do that, we have to explore a little bit. We have to learn a little bit about the circulatory system and how cannabis is absorbed. So first, the circulatory system. There are two complete circuits of the circulatory system. There's two sides of the heart, 4 chambers, and 2 circuits to go with that. The first is the pulmonary circuit, and this is the circuit that brings deoxygenated blood from the heart to the lungs and back to the heart to be oxygenated, and then back to the heart to get pumped to the rest of the body. It's the pulmonary circuit, and it's what's highlighted in color in the diagram right there. The systemic circuit, on the other hand, delivers oxygenated blood to the tissues of the rest of the body. So the left side of the heart pumps that blood out to the upper extremity, the head and neck, and the lower extremity. The thing that I want to point out here that is unique is the liver, because when talking about cannabis metabolism and effects on the body, we have to talk about the effects of the liver. So, unlike any other organ in the body, the liver has 2 blood supplies. It's a pretty amazing organ. Treat your liver right. So, blood that's returning to the heart from the digestive organ, so all the blood that goes to the gut, everywhere from the stomach and spleen, the pancreas, the large and small intestines, and rectum, all that blood, before it goes back to the heart, it has to pass through what's called the hepatic portal vein, the hepatic portal system, to get to the liver. The liver receives about 20% of the cardiac output. So every time your heart beats, about 20% of that blood is going to go to the liver. 75% of that blood going to the liver is going to pass through the GI tract first and pick up nutrients and whatever else stuck in your gut. And then 25% of that blood is going to go directly to the liver through the hepatic artery, bringing oxygen to the liver. So this is a lot of blood. Every 2.5 minutes, a full gallon of blood passes through the liver. It's a pretty massive filter system for the body. All right, so that's going to become important. These 2 sources of blood for the liver. All right, so let's first look at cannabis absorption, and keeping in mind here that for cannabis delta 9 THC to be psychoactive, it needs to be converted into 11 hydroxy THC by the liver. So let's see why it's different when you inhale versus sublingual versus consumption of edibles. So when you inhale cannabis, cannabis, the THC enters the bloodstream really rapidly at the very thin membrane in the capillary beds of the lungs. So you can see where it starts there in the diagram. And it's for the conversion to happen, that delta 9 has to make it to the liver. So let's trace the path that it's going to take. It has to return to the heart before being pumped out into the systemic circulation. And only about 5% of the blood that leaves the left ventricle of the heart is going to get to the liver without passing through a capillary bed. And that's important because when blood passes through a capillary bed, the various cannabinoids that are in the blood can get deposited. They're very lipophilic, and they can get deposited in the tissues in the capillary bed, the cells in the capillary bed. So only 5% of the blood cardiac output is going directly to the liver without passing through a different capillary bed first. All right. Also, vaporizing avoids molecular destruction of smoking, which is why vaping has a longer duration and a more efficient transfer of cannabinoids into the system. All right, so let's compare that to sublingual administration. This is getting into the body very rapidly as well, or relatively rapidly, because it's coming through the very thin mucosal membrane of the mouth, and it's not competing with anything else that's in the mouth at the time, like food that might be in your gut. It has to go through the pulmonary circuit, the entire pulmonary circuit. So it goes from the mouth to the right side of the heart, gets pumped out and goes through the entire pulmonary circuit, goes through the capillary bed of the lungs, and then emerges from that back to the heart, whereby it is pumped out to the rest of the body. Again, only 5% of the blood arrives at the liver from the lungs before passing through a capillary bed, but all of the cannabinoids that were consumed had to pass through the capillary bed of the lungs before making it to the liver. Because of this, because the cannabinoids are bypassing the liver metabolism and going straight into the bloodstream, less 11-hydroxy-THC will be formed than with edibles. And that leads to effects with a similar strength and duration to vaporization. However, if your intention is medicinal and not psychoactive, having a cold extracted tincture applied sublingually is the ideal way to administer that cannabinoid, because particularly if you're looking for delta-9 THC in the bloodstream, because a very minimal amount of it is actually going to pass directly into the liver. So, a higher percentage of the cannabinoids are going to actually enter the bloodstream compared to pyrolysis because of the destruction of the cannabinoids. And then ingestion. So, ingesting the cannabis, the cannabinoids enter the bloodstream right at the intestinal mucosa. And the smaller, more water-soluble cannabinoids are able to pass directly through the hepatic portal vein straight to the liver. And that's 15% of the cardiac output of the blood. That's more than an eighth, about a seventh of the blood of every heartbeat would drive those cannabinoids directly from the point of origin, the short path to the liver. This is a very efficient, this leads to a very efficient conversion of certain cannabinoids to different, maybe more active metabolites by the liver. In contrast to inhalation, all of the active constituents as well enter the body when cannabis is consumed orally, unless an extraction is used of some sort in food. But when you're inhaling the cannabis, you're not actually getting all of the components of, maybe you're leaving terpenes behind, etc. Whereas oral consumption, whatever was in that plant is going into you. There's a second route for cannabinoids into the body through ingestion. So still entering the bloodstream at the intestinal mucosa, but the larger, more lipophilic cannabinoids can get incorporated into chylomicrons and pass through the thoracic duct like other fats. So that's what this extra green line up the middle in this diagram is. It's the cannabinoids start in the GI tract, but pass through the lymphatic system through this, the thoracic duct to the subclavian artery, the left subclavian artery, and then joins the pulmonary circuit, goes to the right ventricle of the heart, passes through the capillary bed of the lungs, where then the blood is oxygenated, returns to the heart for distribution through the systemic circulation, eventually making its way to the liver through the hepatic artery. The thing about this form is these cannabinoids are getting incorporated into the lipoproteins, and that's protecting them from other forms of degradation that can happen throughout the body. This also prolongs the release of these cannabinoids into the bloodstream and conversion of these cannabinoids by the liver. This is really the origin of the very extended duration of effect found in ingested cannabis. Also, that absorption at the gut, at the intestinal mucosa, has a longer time course depending upon what other foods may be present in the gut at the same time. In fact, the quality and quantity of food that's in the gut has a profound impact on that absorption. What other fats were in there, what kind of emulsifiers were in there, how easy was it to assimilate the other fats in that food, etc. How much fiber was there? So yeah, that's a good transition to this idea that the phytocannabinoids are amphipathic. We've seen that word before. It just means part of it's hydrophobic, part of it's hydrophilic. Although they're largely hydrophobic, they're pretty hydrophobic molecules. And if you're going to really incorporate them effectively into foods, they have to be combined with other fats, oils, and then emulsifiers like lecithin that you could find in like sunflowers or eggs or whatever. So yeah, there's a good time for avocados to make a plug for themselves, better than brownies. All right, let's get to a little bit of the food science behind what's happening in edibles. There are two general steps that happen. The first is the preparation of the specific cannabinoid profile for consumption. What sort of physiological or psychological effects? Do you not want any psychological effects? Or are you purely looking for the physiological effects, or vice versa? There's all kinds of decisions that you can make along those lines that affect the selection of specific strains of cannabinoids. The two ancestral strains being indica and sativa, but a host of hybrid strains and different cultivars that have all sorts of different properties. And the question then becomes to decarboxylate or not decarboxylate. That's the question, right? So if you're going to decarboxylate, you are looking for something that is more psychoactive, whereas non-decarboxylation is looking for more physiological effects. So decarboxylation happens by baking the flour. Typical procedure is at 230 degrees or 110 C for about half hour to 45 minutes. And then you can extract the cannabinoids into a fat or oil, either hot or cold, depending upon your decarboxylation goal that you went through or not the step before. The second part is simply beginning to actually make the food, preparation of ingestible food, incorporating those cannabinoids. It can be either eaten raw, if, say, for example, you are looking for the effects of the tetrahydrocannabinolic acid and its anti-inflammatory properties, or some of the other cannabinoids, CBD, CBC, etc. Or the effects of that are variable depending upon the strains that you can select, and there's a host of expertise around that for medicinal purposes out there. Anyways, it can also be used in aqueous or alcohol extractions. These tend to have pretty low cannabinoid content unless they've been concentrated. And that's because it's just hard to extract fats into water, as we know, without an emulsifier. And then there are various extracted cannabinoids into oils that have oils and fats like butter, coconut oil, olive oil, etc., etc. And these have all kinds of uses in culinary applications, either with or without emulsifiers and other lipids that can affect the gastric absorption rate. So you see here on the right, different fats like cheese or eggs, butter, whatever. These are different ways of adding fat or emulsifiers to the cannabinoids to affect the absorption rate. Lastly, I want to talk a little bit about the terpenes. This is an entirely different class of molecule from the cannabinoids. These terpenes, it's a huge category of molecule, and here's just a small subset of various families that are in the terpene family. These terpenes are what give cannabis the distinctive flavors and aromas that we've come to associate with that plant. And a lot of these terpenes have pretty low vaporization temperatures, meaning that they're volatile oils and they evaporate off at pretty low temperatures. This is why cannabis can be so aromatic. So the point is that you can affect the content and the flavor and aroma of the extraction that you make by adjusting the time and the temperature of the decarboxylation and extraction. Also by picking different strains that have a variety of different terpene profiles. There are actually here's just one example of numerous terpene flavor pairing guides. Certainly people are getting into the boutique bud market here by looking at various cannabis cultivars and the terpene profiles they have. All of these terpenes have different physiological effects and significantly different flavor profiles. So, for example, let's just look at one of them, the alpha-pinene. This has an aroma of pine trees, pine needles, wet wood, rosemary, and dill, which are other foods that plants that these terpenes are in. They can aid in asthma, and they're anti-inflammatory, supposedly. And they're in these different cannabis strains with funny names. And the terpene has these different effects like make you alert or focused, tastes good with pesto, pine nuts, rosemary, popcorn, and even wine pairings, pretty sophisticated pairing guides. So all of this just sort of hinting that we are at the beginning of a cannabis culinary boom here as people begin to explore these culinary potentials of cannabis. All right. Well, never thought I'd be giving a lecture on that, but there it is. The culinary side of cannabis. Thanks for listening.